This protocol allows for in vitro evaluation of the impact of confinement and medium stiffness to which native megakaryocytes are exposed to in the bone marrow on their behavior and maturation. The main advantage of this technique is a simplified model which eliminates cell-cell and cell matrix interactions and focuses on the mechanical aspects. Good laboratory practice and sterile working conditions are to be strictly observed. Indeed, even a slight contamination of the hydrogel can have a dramatic impact on megakaryocyte differentiation.
To obtain a single well of hydrogel cell culture, begin by thawing two 1 ml aliquots of 3% methylcellulose at room temperature. Then coat a 1 ml lure lock syringe with the methylcellulose by first drawing 1 ml and then expelling all of it. Next, using the same syringe and needle, draw 333 microliters of methylcellulose from a fresh aliquot. After carefully removing the needle, use sterilized forceps to screw a lure lock connector onto the end of the syringe. Then attach a second non-coated 1 ml lure lock syringe to the connector. Equally distribute the methylcellulose between the two syringes and set them aside. Next, resuspend the previously isolated mouse lineage negative hematopoietic stem and progenitor cells in the concentrated culture medium at a density of 1 × 10⁶ cells per 167 microliters.